Okay. Uh, Sorry for the inconvenience caused. So, uh, my presentation is visible.
Yes.
Okay. So, so this is Dr. Rahul Gupta, cancer and cell biologist, uh, and also a computational biologist expertise. I have completed my PhD from CSR Indian Institute of Chemical Biology, Kolkata, and I am the founder and mentor of OmniBio. My research focuses on cancer biology, CRISPR-based editing, and computational drug discovery. Over the years, I have had the privilege of um, mentoring and training more than 60 students globally, guiding several undergraduate and postgraduate students, even PhD scholars, early career uh, PhD scholars, through their internship and dissertation projects. I have been honored with various recognitions like for Outstanding Poster Award from MDPI Cells, Switzerland, highly commended poster from Royal Society Chemistry London, and the opportunity to represent India as a young scientist at the VetBio Connect International Conference held on Ukraine. So that's a brief introduction about me. If you would like to know more about my research, publication, and achievements, you can see my cursor, right?
Yes.
Okay. So you can connect with for more information about my work and research and publication. You can go through these sites website. This is my personal website, and this is the official OmniBio website. And for future workshops and upcoming workshop notification, you just join this WhatsApp. You can follow me in this LinkedIn profile. This is my LinkedIn profile, and this is the OmniBio official LinkedIn profile. You can also subscribe the YouTube channel, uh, which will provide you free courses videos of some workshops. Okay. So now this workshop is all about learning by doing, from understanding docking fundamentals to performing your own analysis using MJ Doc Tool software powered by PyMol and Discovery Studio. So now we will deep into our workshop. So first we will go through a brief introduction about molecular docking. Okay, what is molecular docking? So molecular docking is a computational method that predict and analyze small molecules like ligands bind to protein targets. Okay, and how it does, it has two process involving conformational sampling. Conformational sampling means when you are putting one conformation of the target and one conformation of the your ligand test compound, the conformation either it will get stabilized or it might get not stabilized, okay, destabilized. So this is called conformation sampling, and this based on these two process you will get a scoring, and this is based on algorithm. Okay, so the algorithm, what it does, it explores the conformational space of the ligand within the protein binding site, okay, and generate multiple poses that represent different binding orientation of your ligand at the protein pocket. Okay, each pose is evaluated by different score, and this scoring is is done by the help of interaction studies. What are the kind of interaction studies? The most most prominent interaction studies are hydrogen bonding, okay, and then after that it is hydrophobic interactions like electrostatic interaction, van der Waals forces, and finally it based on the entropic process, which is called as binding affinity process. Okay, now as previously the docking process was very slow and it takes huge time, and we can do docking one by one with your ligand, but now you can do docking with thousands of compounds simultaneously. Okay, making this virtual screening or computational method a powerful tool to identify promising drug candidates. So what is the advantage of molecular docking? It is a cost effective virtual screening, because see, if you will screen thousand of compounds, you will purchase the compound and then you will test each compound in the wet lab, and you will find that thousand out of thousand of compound, one or two compound is giving you the activity. Okay, it's a active compound, but for that you will need lot of money and lot of reagent, lot of chemicals, and so instead of screening in wet lab, the researchers and scientists have developed this in silico process to save the money. So it is a cost effective virtual screening where we can screen 1000 or 10,000 of compounds by creating a library, and out of 10,000, for your particular target, which compound is active, you can find it out. So it's a not a validatory you cannot tell that, oh, this will happen. Okay, it's just a predictive method. For validation you have to perform experiment, but for see it brings thousand compound to suppose one to five compounds, so your money is getting saved. Okay, instead of screening thousand, you can screen now one to five late compounds, which are the hit compounds you get from this molecular docking process. Okay, it's a rapid and evaluation of large compound libraries, what I explained just now. You will get the molecular insights which will not get with the wet lab. What interaction it's playing with it, okay, what is the binding affinity, what is the energy minimization and requirement. So all these you will get through in silico method. So now what is the disadvantage. See, in physiologically, the protein remains three-dimensionally and it's flexible. Even whatever the compound, or suppose you have taken drug, that drug all will also exist in 3D conformation, and it will also have some orientation or rotation. Okay, that will be also flexible. But as we are mimicking the physiological uh, body to a computational method, so obviously there will be a limitation. We cannot give that much flexibility to the protein structure or the ligand, so that it can act like a normal body physiological uh, presentation of that structure in computation. So that is the limitation. Okay, the protein flexibility will be less, and further ligand conformation will also be not that much flexible. So this is the disadvantage. But for that, scientists have developed that molecular dynamic, in which both protein and ligand is in the biologically related to orientation, and it's in dynamic in nature, means it can be rotate itself accordingly, similar to the body physiological process. But still our body has lot of uh, requirement, like what water is there, interaction is there, other enzymes are there. Okay, so you can't give that one in computational method, so that is also having some drawback. But it can somehow mimic near to the body physiological condition. Okay, so next is scoring function inaccuracy. Okay, sometimes you will find like D glucose and L glucose, if you will dock to a glucokinase protein, it you will see that D glucose is having less docking score, while L glucose is having higher docking score. Okay, but as we know that L glucose is not biologically active, it's not functional, but D glucose is biologically functional. So in that case, you will see that scoring function has having some inaccuracy. Okay, now difficulty handling like induced fit effects, uh, the interaction with where the your ligand will bind to the pocket of your protein, that is, we are giving some target site or like in the form of coordinates. Okay, which is a binary system, an algorithm system. So in like that we will induce the ligand into the pocket of the target protein. But in our body, it is not like that. It can interact in the target of the protein, even it in the different site of the protein, and it can change the biological function of that protein. Okay, so that is another challenge, that it is not getting completely induced fit during the docking process. Now the next challenges is like metal coordination. When we will do the docking process, we will find that we have to remove these all the cofactors and ions present in the protein to get, so that we can get the ligand interaction only, not the interaction with these metal ions. Okay, now what are the applications? uh, we application is that like drug discovery and lead optimization. We can discover drug based on the structure. If you have a novel compound, you will find for that compound you will find a target, and you will screen thousand of target, and you'll find the perfect target. Suppose you have taken one novel compound and you have screened for thousand of target, like target protein, acetyl coenzyme is there, glucokinase is there, and then protease is there, mmp9 is there, several proteins are there. But you will find that this your novel compound or the compound which you have which you wanted to test it, it's binding more effectively to one of the target. Okay, so in this way you can screen your drug, and you will do the drug discovery, and also you will do the lead optimization. Okay, then you can design inhibitor for enzyme with after binding to the enzyme, that drug or compound which you have developed is inhibiting that enzyme or not by this protein interaction. Now toxicity prediction we can do, that is called ADMET analysis. Toxicity you can do toxicity prediction, like thousand of ligand you have taken, all the compounds will not be biologically good for your health. Okay, it may have some oncogenic properties or some uh, allergic properties which will which will be predicting through this ADMET analysis. So nowadays AI has been introduced in most of the uh, scoring functions. Okay, through Python algorithm. So this is enhancing the scoring function inaccuracies. Okay, now dynamic docking methodologies is there. Okay, which I have explained, that molecular dynamics can mimic the body physiological condition. So this is the brief introduction of the molecular docking. So now there are different types of molecular docking. Okay, the first is the rigid docking. Rigid, yeah, as the name suggests, that both protein and ligand will be as fixed structure. Okay, so in this case, the computation requirement is very fast, but it is limited. It's not accuracy is very low, and it is considered as initial best for initial screening of large compound libraries. It's, for example, if you'll see HIV-1 protease inhibitor designed through this rigid docking process, because the active site remains constant across different bound state, so making its rigid docking highly effective for initial screening of potential inhibitor. Now mostly we used to perform this semi flexible docking, and in this workshop also we will do this semi flexible docking, in which ligand remains flexible while our protein is rigid. Okay, and this balances computational efficiency with improved accuracy. Okay, like in the rigid docking you have your it is fast, it is faster in computation, but this is like it is balanced, and it has good accuracy in predicting the bindings than the rigid docking. Okay, then the next is the third is the flexible docking. Okay, this flexible docking, in this flexible docking, it the both ligand and protein side chains or backbone are flexible. Okay, like in our like similar to our body functions. Okay, so now computationally it is intense. It requires a huge computational process, and it requires supercomputers and high speed internet for for working. Okay, so it also captures induced feed effects, okay, and conformational changes upon binding. For example, if you will see the kinase inhibitors which have been developed, it requires flexible docking. Why? Because this ATP binding site, most of the kinases have ATP binding site, undergoes significant conformational changes upon ligand binding. Okay, so once the ligand binds to their binding site of that kinase in the ATP binding site, the conformation changes. Okay, it varies to ligand to ligand. Okay, so so flexible docking is very good in that case. Okay, now we will compare the analysis of docking method, like which one is good and which one is better, and which and it depend upon your docking choice and your re- computational resources. Okay, so rigid docking, protein flexibility is, as I told, it will be no flexible in rigid docking. Protein is not flexible. Computational cost is low and accuracy is moderate, and it's faster. Semi flexible docking, which we will perform, this side chains of the protein is all remains only flexible, ligand is flexible. Okay, computational cost is medium and accuracy is high. Flexible docking and ensemble docking, which comes under molecular dynamics, the protein and ligand both has full flexibility, computational cost is very high, and accuracy is very high and also excellent. Now, what are the general steps in molecular docking? This is the most important part of our workshop. So, listen carefully. So, first is the protein preparation. For that, what we have to do, we have to first retrieve the PDB. Means we have PDB, PDB is the protein dat, data bank. Okay, where protein different protein experimentally derived structures are being stored. Okay, so first we have to retrieve the protein from PDB, then we will remove the waters. Okay, as crystallographic structures has some water, so we have to remove that water, and then we have to add hydrogen atoms, then we have to optimize side chain conformation, and then assignment, we will assign the partial charges, and we will include cofactors and metal ions, which is essential for binding, and then we will validate our structure quality using Ramachandran analysis. Now the second step is for ligand preparation. We will generate first 3D structure. Either we will download it from SDF formats, or we just uh, see, copy the smiles which is present in the PubChem or database, or drug bank database, or zinc database. Okay, then we will do energy minimization with providing appropriate force fields like MMN 95 and GAF, okay, amber. These are the different uh, eh, minimization of energy force field program which is being used in the software. Then we will consider enantiomers. Enantiomers means the compounds can exist in different forms. Okay, because due to the arrangement of the or ionization of the compound atoms. Okay, so then we will validate the stereochemistry of verification, where that is we will generate a clean ligand. Then then we have to define the active site where this ligand will bind to the protein, target protein. So that site is called binding site or active site. Okay, or active pocket. And how we will do this? Either we will do through a literature review or through a computational methods. There are several sites where you will put your protein pdb structures in the pdb format, like cast-p is there. Okay, pranked web is there. So they will predict different pocket according to the conservation of the conservation residues present in the pocket region. Okay, they have that algorithm, and they will recognize that, and they will give you the pocket coordination and residues which need to be target. Okay, and for that we have to define grid boundaries that is present using the software, docking software. And we will define the binding site. Then we will do a docking, and then after docking we will do docking calculation. Okay, we will uh, calculate the docking based on the scores, docking scores. It will provide you docking scores and different poses after binding of the ligand to the target protein, and it will rank based on the scores. So now the next will be the result analysis. In that what we will do, we will evaluate different binding poses on scoring functions, and then we will check the chemical interaction. Okay, after the chemical interaction we will analyze this data through uh, analyze this data, and we will validate it if it's necessary. We will validate. What we will do, we will get the result, and then the next step will be validation. What we have to do, we have to perform that experiment on wet lab condition. So that is the way to validate your computational methods. Okay, so now for validation, we can do, we have to design the biochemical assays to predict. Like we can do mtt assay for cell proliferation to check your compound is actively inhibiting the cell proliferation of the post cancer cells. Okay, so like that we have to design our experiment for validation, which will so our part will be protein preparation, ligand preparation, active site definition, docking calculation, and result analysis. Okay, and then we will generate images like a publication. Okay, ready to publication images we will generate from after analysis using pymol and discovery studio. Now what is protein? Okay, we and why it is called as receptor. So in so the protein target is the receptor. What is that? It is a macromolecule. It can be an enzyme, receptor protein, or transporter protein. Okay, to which this drug means your ligand will bind to exert its therapeutic effect. So what is protein? So proteins are large complex biomolecules made up of amino acids linked by peptide bond. Now they have four general structural organization, which is primary structure, secondary structure, tertiary structure, and quaternary structure. Primary is the just linear sequence of amino acids arranged. Now secondary structure is is the local folding and generating alpha helices and beta sheet type of structure is called secondary structure. Now tertiary structure is what? This helix and beta sheet and loop will join and it will generate and it will fold into a three-dimensional structure in a single polypeptide chain. It will generate a tertiary structure. Okay, and this tertiary structure is the functional protein. Okay, this tertiary and quaternary structures are functional protein, and the quaternary structure is what? The assembly of different or similar polypeptide chains together. Okay, see, this is one tertiary structure. So again one more tertiary structure, and it will link with one loop, so it will become a dimeric protein. This quaternary, this is also called as quaternary structure. Okay, and these two tertiary structure and quaternary structure is the functional protein structure which provide function to the protein. Now where to get this protein where we will get this protein structure for our molecular docking? The main source is the protein data bank. Okay, so what is protein data bank? The protein data bank is the central repository of 3D structural data of proteins and protein ligand complexes. Okay, now the primary repository of experimentally determined protein structure contain over 200,000 entries in this protein database. Now there are different protein database like RCSB PDB is facilitated by USA, and it is another one is from Europe, and the third one is from Japan. These all are partners. They shared the information with each other, but the central one is the RCSB PDB USA. Now each structure of the protein is identified and given a 4-character code. Like example 1A to C is the acetyl coenzyme. Okay, and like 5OQW, it's a ZIAP protein, means X linked apoptosis inhibitor protein. Okay, now these protein structures is determined by different experimental methods like X-ray crystallography, NMR spectroscopy, or cryoelectronscopy, or they can be identified with the combination of these three experiment experimental experiment separately, or it can be individually determine the structures. Okay, now there is the selection criteria. How there are a lot of PDB structures stored in the protein data bank, but which protein is best for your molecular docking process? So what we will there is a selection criteria. The structures with resolution which is less than 2.5 angstrom for X-ray structure or cryo-EM structure, the resolution of the protein must be lower than the 2.5, and similarly the R factor values must be lower than the 0.25, and R free value will be 0.30, and these two factors, resolution and R factor, determine how better is your model of the protein, and this also determine how your protein conformation is in best condition, and how the atoms and the residues the of that protein is arranged very nicely in coordinated way. Further, the structure should have minimal missing residues. You will find some of the proteins when you download it, and you will see in the visualization tools like Discovery Studio or PyMol, and when you will check the sequence of that pro, of the protein, some proteins will have some missing residues. If you will do docking with that missing residues, and if the active site is present in that missing part, so you will not get your result. Obviously your docking process will consider as a failure. Similarly, uh, so you need to have minimal missing residues, or no, or complete structure. Okay. Now fourth criteria is the ligand always look to the target protein having a co-crystal ligand, so that you can get your binding site information easily for your target, so that you can target your hit compound to that particular site where the co-crystallized ligand is bind, because you know that that is the active site, and already someone has performed that experiment. Okay. Now, and the biologically, the structure of the protein must be in oligomeric state, which is, I said, that tertiary structure, or in quaternary structure. It must be physiologically in active state. Okay. Now there are different alternate protein database. Uh, you can go through and check these databases. In like in Uniprot, you will get different the comprehensive protein sequence and functional information. Okay. Now NCBI protein database has curated protein sequences. You will get the protein sequences from gene bank. Swissprot is is a curated form of Uniprot with annotate manually. The protein structures are annotated manually. Okay, like now PFAM is used to determine protein families and domains. Cake pathway is to determine proteins and metabolic and signaling pathways. String database for protein-protein interaction. So there are different protein databases with different functions. Since we don't require this in this workshop, so we will only go through the PDB database, that is protein data bank, to retrieve our protein. Now see, now we know that we need experimental structure. We can do this docking by downloading the experimental structure from protein databases. Okay. But the protein structure, or the structure which are not available in the experimental form, so for that what we have to do, we have to perform homology modeling. Okay. And this homology modeling is essential, again essential, to define the protein structure which lack experimental determination. Now there are different softwares where you can perform this molecular homology modeling, and it requires various steps, like we have to download the sequence, and then we have to find the template, and then we have to, after template, then we have to search and form a model, and then we have, after predicting a model, we have to validate the model. Okay. So it requires another workshop. We will not be doing this homology modeling in this workshop. For that we will do a new workshop where we will see how to develop homology modeling, how to find the binding pocket size, and how we will perform the docking with blind docking and also with the targeted targeted or side directed docking. And the software which usually used for uh, developing this homology modeling is Modeler, Swiss Modeler, iTaser. So these are the different tools. Now AlphaFold is the best tool to predict structures, homology structures. Okay, and it also has now introduced an AI feature. So it will detect your uh, protein sequence. If you will provide protein sequence, it will give you the best homology model structure. And as you know that in 2024, in the Nobel Prize in chemistry was awarded due to the development of this AlphaFold database, which is AI based. Now it is an AI based tool which predict the protein folding and the structure, 3D protein structure near to the experimental accuracy. Okay, so till now you are clear with this. You can see this is the figure I have generated. This is my work where I have generated the homology protein structure through AlphaFold and Swiss model, and then then I have validated it, and then we have find the functional domain which is present in the this homology model structure of this protein, and we determine the pocket site, and you can see it is giving a confidence score, and we used to screen the homology modeling homology modular structure with this confidence score, q score. So our q score is 90.76. So greater than 85, greater than 85 score, q score considered to be the best or considered to be the better model. You can go for the docking process. So now we will do some hands on. So for that, what we will do, first we will download the protein structures. Okay, so for that we will go to the protein data bank site, which is www.rcsb.org, and from there we will download our target protein in the PDB format. Okay, as I have told that we will also check the selection criteria. Okay, so till now, students, please respond. You are clear? Hello.
Yes, sir.
Okay. So now we will proceed with the hands-on training. Are you ready?
Yes, sir.
Okay. So I am sending this link in the chat box so that you just copy and paste in your web browser. Okay, just copy this and paste in the web browser. So this interface will come, which is the protein data bank interface. Are you people doing this? Hello.
Yes, yes, we're doing it.
Have you opened this site?
Yes,
Okay. Now you just type here 5OQW in the search button. You can find this search button here in the right hand side. Okay, and there you have to type 5OQW and then press on the search button. Do you get this structure and this 5OQW protein in this working space? Have you received that after searching? Hello. Are you people following?
yes, we're following. We're following.
See, again I will tell you what we have to do. We will go to the browser and then we will paste the address which I have given in the chat box, and we will open this site, PDB site. In the right hand side you will get the search button. Okay, it's clearly it will be written like enter search term, ligand id or sequence. As from the theory I have explained that each protein has its own 4-letter code. So the 4-letter code for our workshop which we will perform docking throughout that protein is the code is given like 5OEW. Okay, and then press enter. Once you will press enter, you will get this page where you can see the structure of your protein. Okay, this is the structure of your protein, and what kind of protein it is. It's a apoptotic protein. Okay, which initiate the apoptosis by binding to the caspase executioner, which execute apoptosis. Okay, it's a see the experiment who have the research the researchers who have experimented on this protein is will be given in the literature. Okay, this is the work they have performed. Okay, and this ASTX660, which is also known as A4E. Okay, this is the inhibitor of this protein. Okay, this ZIAP protein, and this ZIAP protein is what is the inhibitor of apoptosis protein. It, so you can follow this paper and you can see the work of there. Okay, so this is the POI of the paper. You can click and you can download this paper, and you can use as a reference for your work. Similarly, for if you search any other protein, okay, like any other protein, it will see the same thing, it will appear. Okay, Now, as I now we will come to this our targeted protein. So transcription of the protein is apoptosis. Organism is Homo sapiens. Okay. Now the protein has been co-crystallized by using expression system, which is developed in PL21 E. coli cell. Okay. Now selection criteria, as I have explained the selection criteria, the the resolution of the protein should be less than 2.5. So you can see the resolution of the protein is 2.31. So it's a good defined structured protein. Even the R values are less than 2.5 and 2.0.
I'm okay.
2.3. Please unmute your mic.
Yeah, yeah, no.
Hello. Yeah, thank you. Uh, So now what? Now we will go scroll down. You can see this micromole content tab, where it will be given the structure. What is the molecular weight of your this protein? Is the molecular weight of the protein is 30.39 kDa. Okay. Now how many residues are there? Their residues count, and how many residues deposited in that. Okay, you can see that information. Now if you will scroll again in the down, so you will find that there are chains. There are two chains. Obviously it will be a two chain, because you can see that the green color is the chain A and the orange color is the chain B. Okay. So this is the dimeric form of this protein. So this, even the if the protein is in monomeric form, it will be active. Okay. And even if it is a dimeric form, it will be active. Okay. Now if we'll click here in structure. Yeah. So this is the whole protein. Okay. And you will see this small dots are what? It is the water molecule. And if we'll rotate, click click right button and just drag to make it align to the center. And now click left button and then rotate it. Move right, left and right. You will see that your the protein is, you can rotate. Once you will rotate, you can see the ligand. Okay. This is the ligand. You can see this is this, as this protein uh, having a two chain, so the ligand will also be two. Okay. Because the ligand is binding to the pocket side. So this is the ligand for chain A, and this is the green color one, and the orange color one, this is the ligand for chain B. And this ligand is what? That A4E is the inhibitor of this ZIAP protein. Okay. So you can zoom out. You can zoom out by just if you are using mouse, then you can use your center scroll button to zoom in, zoom out. And if you are doing laptop, then use two finger in your mouse pad, and you can move up and down. You can see the protein is getting zoom in and zoom out. Okay. So from here you came to know that it is a dimeric protein having chain A and chain B, and the bind and each chain has its own binded ligand. Okay, further if you will see in here in the sequence, the sequence of the protein will be provided. Okay, and this sequence, if we'll copy this sequence, and you can keep this sequence for developing, if you want to develop your own homology modeling protein, then you will use this sequence. Now there are different other tab, okay, which will tell you about annotation of this, how they have annotated the protein structure, labeling, and what how they have done labeling, and how they have classified. Okay, all these, what type of protein, what type of domain is there, okay, what type of protein family is there, gene ontology, all these you will get in annotation. And if you click on experiment, you will see how they have perform the experiment to crystallize the structure, what method they have used. Okay, all the data will be present for for developing this protein structure using X-ray crystallography. What software they have used for determining the crystal structure after forming crystal structure of this protein. Okay, then in sequence you will get the whole sequence of the protein. Okay, and you can, if you click on genome, it will show the site where the sequence of the nucleotide sequence of this protein residing in the chromosome. See, it's reside in the, you can see it's reside in the chromosome P25 region, Q25 region, chromosome X. Okay, it will show you the location of this protein. So these are the information you can get. And what type of ligand is being binded, that is we called as co-crystallized ligand. See, this is the name of the ligand binded to the this target protein 5-OQW, which is ZIAP protein, and the name of the short name of the ligand is A4E, and this is the chemical name of the structure. Okay, so here you will get the ranking of goodness of fit, means how this ligand A4E is binded to this targeted protein. So if it is above 55% or 50% ranking for goodness of fit, then it's a good ligand. We will consider this ligand to be active. Okay, so now we will go again back to a structure summary. You will scroll down to the macromolecules, and you will see the sequence length is given is 127. It has chain A and chain B. If you will scroll more down, now what are the small molecules present in this crystal structure of the protein? See, this is the ligand A4E. Okay, they have annotated as E and G for chain A, and for chain B it is annotated as G. Okay, similarly this zinc ion is present. This zinc ion is required for the activity of this protein. So during the crystallization they have crystallized this zinc ion, but for our molecular docking process we have to remove this zinc ion, this sodium ion. Okay, so that we can only see the interaction happening between the ligand and your protein. Okay, so this is the experimental data validation. So this is about PDB. Now what we will do, now we will download this protein. Okay, now we will download this protein structure. So go to download files. In the right side, right corner, you will find download file option. Okay, and then you have to click in that. In You will get one pop-up box. You go to the legacy PDB format. Okay, click in this legacy PDB format. It will The protein structure will start. It will start downloading. Go to download to check. It's not started yet. Get some gateway error. Okay, I will try. Have you people have downloaded? Hello.
Yes, yes.
You people have got this protein structure downloaded.
Yes.
So, so no issue. I have already downloaded this structure before. So no issue. Okay. I don't know why it is showing error again and again. Okay. I have. Oh, okay. So this is the way we can retrieve our protein structure through PDB. Again, I will try. I think server is too busy. No, it's very slow. Usually it takes very few seconds to load and okay, no issue. I have this protein. Can go to download and yeah. So it's there. Okay, you can see this. I have downloaded this protein earlier. So what we will do, after downloading, you have to do what? You have to copy this, and then you have to create your working folder in the desktop. named as MZ Doc Workshop, and now you just make one more folder inside that MZ Doc Workshop folder in your desktop, and then inside that you just create one new folder and name this one as files for docking. Okay, now I will move that downloaded protein to this working directory file folder, working directory folder. Okay, so in this way we can retrieve or download the structure of the protein, which will which will use for docking process. Yeah, see, in previously this part is been not get it loaded, so I couldn't able to explain this. So this is how they have done validation, or how you will know. The main part of this one is you can see R free score. See, R free score is very less. Okay, so it's a good protein structure, and the main analysis is the Ramachandran outliers. Okay, you see if you'll remember the Ramachandran plot, I will show you in my PPT. Yeah, see, you can see this is the Ramachandran plot. This is the 2D way of representing the Ramachandran plot, and this is the 3D way of representing the Ramachandran plot. This Ramachandran plot is used to determine the protein conformation residues of the protein conformation present in the allowed region, and non-allowed region. Okay, and it calculates the phi and psi bond angles. Okay, it starts from minus 180 degree to 180 degree, and it also signifies that the residues are in the orientation, or it's in the best conformation, which is making this structure stable. So typically if the structure is the outlier of the structure is less than 5%, and 95% of the residues are in the allowed region, then the structure is considered to be the best model structure. So if you see this 2D plot, you just divide into four quadrants. Okay, this is the first quadrant, this is the second quadrant, this is the third quadrant, and this is the fourth quadrant. Okay, the first and fourth and the third quadrant is, if the residues are present in there, you can see this red dot, most of the residues are in the allowed region, and this is the second quadrant. Okay, this is the second quadrant. uh, this quadrant is considered as the disallowed region. Okay, mostly proline and glycine appears here, because this glycine and proline are rigid, they don't have any alpha carbon, chiral carbon, and it does not have any rotational bond, so it is very strict. So if the protein is having more number of glycine and proline, it will obviously comes in the disallowed region, and the protein will not be considered as a good protein. So most of the, if you will see, most of the protein does, if you check the sequence, there will be very less number of proline and glycine sequence in the protein structure. So this Ramachandran plot is used to determine the structure which are stable and stereochemically confirm conformer of the protein structure. Okay, so this is uh, this is the way you will select your protein. This is the selection criteria to select your protein structure for docking. Okay, now what is that we have to know the ligand. What are the ligand database is there, and what is ligand? Okay, so we have completed the retrieving the protein structure. Now we will know learn about ligand database and how we will retrieve it. There are different database present in the uh, web server. We can use public database. These are public database. So this one database is ChemBL database, PubChem repository, drug bank database, and Zinc database. Mostly this Zinc database and PubChem repository database is being used to retrieve different compounds or molecules, and Zinc database is mainly used to isolate drugs which are already available or and validated. For PubChem, you will find some new compound, and even some old compound and validated compound and non-validated compound also you will get in PubChem. So what are the different steps to retrieve these databases from ligand from these databases? We will use PubChem as the database to download this ligand. So I will copy this uh, link website of the PubChem, and I will share with you all in Google Meet chat. Yeah, okay. So use this link to open this open the PubChem. Have you opened this site, PubChem site? Hello, hello, please, student, kindly respond. Have you opened this site, PubChem site?
Yes, yes.
Okay. So now we will download our ligand. Okay. First we will download. You type mebendazole, 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 and then you just click on search.
Can you place the name on this in on the
What?
comment section? Help us place the name of the ligand on the comment section. You can just copy and paste it now.
Okay. Okay. Sure. these are the this compound is anti-helminthic compound, okay, used to treat parasitic diseases. You can but it's been used as a repurposing drug for cancer treatment. And this drug I have been used for repurposing and for ovarian cancer in my PhD research work. And you will find that this drug is very effective than most of the cancer drugs which are present in the market. Okay, this mebendazole I have given the name of the mebendazole in the chat box. You just copy and paste in the PubChem search bar and just press enter for searching. The first the first name of the compound, it, you just it will appear, you just click in that, okay, and then the best match, you will get the best match. The first one is the best match. You just need to click here, click in the name of that, name of the compound. It will open. See, you will get the structure of this compound and all the information from, but we don't need the information, uh, all this information. If you will write, but if you have a novel compound or different compound, as I know this compound very well, so I don't need to read all this information. But if for you this will be a new, so you can read and you can know what type of compound it is. Okay, so the molecular weight of the proton compound you will get from, you know, then what are the groups present in this compound, what type of compound it is. It is a benzimidazole compound. It is having a methyl and benzimidazole carbamide group, benzyl group in the fifth position. Okay, and similarly you will get the 2D, this is the 3D, you will get a 2D structure, and then you will get to see 3D structure, crystal structure also of the of this ligand, and then you will get this uh, key key to key nomenclature of this compound, and this is the IPC name of the compound. Similarly you will get the Kell nature of the compound and smile of the compound. This smile is important. You can copy this smile, and also you can use this smile when you will use for docking. You just put this smile in instead of uh, the SDF file which we will download. Instead of file, you can use this smile of this compound, where if you, when we will perform docking, it where it will ask the smile, give the smile of the compound. You can use in that case. Okay, so this is the way you can go through any compound you want to know its information. You can go through this, mm, through this PubChem database. So now we will go to download option, which will be present at the right hand side. Just click in this download. One dialog box will appear where it will, where you can see different files, file format present in the this box, and you can download any of these, any of the file. But mostly for docking purpose we will use SDF file. And even we don't use 2D structure confirmation, we have to use 3D SDF conformation. Okay, if you will use 2D structure conformation and you will do the docking, you will get the docking result, but the interaction or because the interaction will not be happening in the, happening it will be in planar structure, so the interaction, different interaction will fail, while in 3D conformation, as the structure is in 3D, it will completely dock with your target protein in the pocket site. Okay, and the interaction will be much better than the 2D structure. So what we will do, we will download this 3D conformer. Okay, we'll go to the SDF and we will click on save. As you click on save, see it will start downloading. So now once downloaded, you just click show in folder. It will take you to the download option where the file is getting downloaded. Okay, now you will copy this file and we will move this file or copy this file to our working directory. Okay, got it. So we have protein structure and we have ligand, target ligand. Now we have to also download the co-crystallized ligand to validate our docking process. How we will validate the whatever docking what we are performing and the result we have got, it is reliable and it is reproducible, whether other can also reproduce or not. So and whether your docking method is correct. So to validate it, so we will use co-crystallized ligand as a hit or your test compound, so that we will know it is we will know that whatever the co-crystallized ligand is bind to the pocket site of the target protein. Similarly, after docking, after performing your docking process, this the ligand which you have downloaded, okay, it's also binding in the same way as as the co-crystallized ligand is binding to the target protein. Okay, so for that again we will go to pubchem. We have to open again that pub pdb pdb to pdb and you have to open the pdb again. Again we will search 5050qw and we will go to the ligand button and we will copy the name, this chemical annotation of the compound. We will copy it and now we will go to the pubchem and paste this one in the search bar and then we will search. Again the best match, you will get the best match, and the first one is the best result, and this is the co-crystallized compound which will need to do docking, means we have to do redocking to validate whether our docking process is correct or not. So click on this, again go to download, go to 3D conformer and go to SDF and click on save. So it will start downloading. See, download is completed. Again we will click on show in folder. So before moving, as see which the drug mebendazole which we have downloaded, it has given some ID name, okay, ID name, and even the compound which we have downloaded, this co-crystallized compound, is also having some ID. It looks similar, so it will make confusion. So what we will do, we will rename. First, before moving to the working directory, we will rename. We will write the name of this ligand. What was the name of the ligand? It is A for A4E. Okay. Rename A4E. That rename conformer. Yes. Okay. Now we will copy this and we will paste this in the working directory. Okay. So as we have this, now we have co-crystallized ligand and we have our target ligand and we have our target protein. So now we will also rename our target or hit ligand, target or hit ligand. I will give short name this, as this drug name is mebendazole, so we will use the short name of this drug MBZ. Okay, MBZ for mebendazole. Okay, we will also remove this number. We will also rename other file also. We will remove this number and just we will keep co-crystallized ligand as A4E. Similarly, we will do rename the protein file also, and we will just keep 5OQW. Okay, till now everyone is clear? Hello. Hello. Have you saved these files in your working directory? Hello.
Yes, I'm here. I'm here. I'm trying to download the second inhibitor. Ligand is not working. I've not got it.
Co-crystal
Trying
ligand.
to get the second ligand.
The co-crystal ligand, you didn't got it. Peter, which ligand you have not got?
The co-crystalline ligand, the one from the PBD.
Okay. I will paste the name of the name of this co-crystallized ligand in the chat box, so that
Okay, can you help me?
you just
Can you guide me how to get it?
yeah, I'm telling you, you just I'm sending you the name in the chat. I will copy the, see, I have copy the and given you the name of the co-crystallized ligand. You just copy this, go to pubchem, and then paste this in the pubchem search bar and search. Okay, are you following? Hello.
I can hear you. I can hear you now. My network was a bit bad, but I can hear you now. Okay, let me copy it on.
Have you copied that
Okay.
name?
Yes, yes. So I should paste it in PubMed.
Now you go to pubchem.
PubChem, okay. 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 Let me check for PubChem. Okay, I paste in PubChem.
Yeah, now search it.
Uh,
You can see one magnifying glass like button.
yeah, yes.
Search it or
Yes.
press enter. It will automatically search.
I'm searching. Okay, I'm searching now. Let me get it up.
Okay. Once you will search, okay. After searching, you will get this best match, and you will get the first compound name. It will appear. You just click in this one.
Yes, I'm there now. I'm there.
Click in this one and just go to download and click on the download.
Okay.
You will get this dialog box.
Okay. So which one should I download?
You go to 3D confirmer.
Okay. Let me just
Go
do
to
that
3D
now.
confirmer and go to SDF and click on save.
Okay.
Hello, Doc. Doc, good afternoon, sir.
3D conformer.
Go to 3D confirmer, go to SDF.
SDF. Okay,
Yeah,
save. Okay, thank you.
SDF and click on save. You will see it
Yeah,
will
saved.
start downloading.
Yeah, we got it now.
Once downloaded, you just move that, rename that file. First rename that file as A for E.
A4E.
Yeah,
Okay.
A for E. That is the name of the co-crystallized ligand in short form.
Alright.
Okay. After renaming it, you just move that file to the working directory.
Yeah, I does it. It's okay.
Done?
Yeah, done.
Okay, good. Anyone is
So I
having
have
any
three
question?
files there.
Yeah, tell me,
I have
Peter.
three files in there now. I have the protein, the mebendazole, and the A4E.
Good. So this file we will use for the docking process. Okay.
Okay, thank you so much.
Okay. So I think your problem is been solved, Peter.
Yes, yes, yes.
Okay. Anyone is having any problem? Please respond. You can unmute your mic, Peter.
Okay, okay.
Please respond, student, so that I can proceed. Hello. So till here we have completed. We have so we will just recap it. So we have known what is molecular docking, what are the advantage and disadvantage of the molecular docking, what are the application of the molecular docking, and what is the core principle of the molecular docking. Then different types of molecular docking, what is rigid docking, semi-flexible docking, and flexible docking. We have done the comparison and which one is the best choice for your docking. Then we have gone through general steps which will be requiring for molecular docking in the protein preparation, ligand preparation, active site generation, docking calculation, and result analysis. Then what is protein, what are the different structure of the protein, different form of the protein, and what is the functional form of the protein. We have discussed about protein databases. Okay, mainly protein data bank from where we have retrieve our protein structure for this talking workshop. What is the selection criteria? How we have select? How we have to select the target protein structures and download from this protein data bank. So we have came to know with this selection criteria. Then other protein database, and then if you don't have experimental structure or the structure is not present in the protein data bank, what we will do, we have to perform homology modeling. Okay, so homology modeling we can know, we can know the structure of the unknown protein, and we can perform molecular docking in the similar way. Now we have follow the steps to retrieve the protein from protein data bank, and we have already downloaded our targeted protein. Then we came to know about ligand database and how we will retrieve the ligand or chemical compound from the pubchem database, and which format we have to use. So so for protein, the format of the file will be .pdb, and for molecule, for ligand, the format of the file will be in 3D conformation, and it will be in .sdf. Okay, so this is it for the first day of the session. Okay, so now I will tell you what we will learn on day 2. Okay, so first will be the introduction to the MZ doc. Okay, and then we will know the steps which are involved in the protein preparation and ligand preparation. Then, uh, then we will know how we will visualize the result and in Pymol and Discovery Studio. How what is Pymol? uh, Pymol brings in the docking process, what Discovery Studio being used in the docking process, how we will generate publication ready images, and then as I have told, the registered student will get extra bonuses. That is, I will tell you how to write this result uh, interpretation using AI tools. I will give you ready to use chat GPT or prompt which can be used to write this result and interpret this result using this AI tool. Then I will tell you the tricks, new tricks which have been uh, introduced in PyMol and Discovery Studio for visualization and generation of different images. Okay, and this is for registered students. You will get these bonuses in this workshop beyond learning the molecular docking, and you will, as MZ dock is the new molecular docking tool, you will and it is a next generation docking tool with automation system, and it is having, and it worked more better than the PyRx and AutoDock, because its Mina is the docking is core, which is being used for, which is being used in this MJ dock, which is far better than the scoring system which is being used by the PyRx and AutoDock. So you will be learning this new tool. As you can see, you can do multiple docking also, as we have downloaded two ligand, so we will also perform multiple ligand docking. Okay, then, and after registering, what other thing you will get, you will get this day 1 session recording and day 2 link, certification of participation recognized by OmniBio, and one of our partner is the Reach Wave, and you, and also Biotech Wala is there. That is also our partner in this workshop. You can follow Biotech Wala also, and we will have some assignments and quiz to test your understanding, and we will have one-to-one doubt clearing session after the docking workshop, and the group will be active for the lifetime. Publication ready and molecular images template will be given. Okay, and how you will write the paper, that will also you will get to know. I will explain that part also. So so be ready to get these hands on, and I will share some links. Thank you to join in the day 1 session, and for before ending, I will give you some links in the chat box. First, I'll give you the feedback form of today's class. This is the feedback form. You need to fill the feedback form. All the student must fill this. It's compulsory. This is the quiz. You need to practice this and answer all the questions so that I can analyze your knowledge, and whatever you have learned in this workshop, I can analyze you people. And the second and the third form is the assignment, so that you will practice whatever you have learned today in the best way. So these are the three forms. First one is the feedback form, and second one is the quiz form, and the third one is the assignment form. Okay, these are the Google form which you need to fill and answer it, and upload this till 3 p.m. Tomorrow, and submit this one. Submit your responses, okay, till 3 p.m. tomorrow, and we will start the workshop around 6 p.m. There will be shift in the timing. Okay, it will be like 6 to 8. Okay, and kindly join earlier because tomorrow we will have a lot of work, and we will learn a lot of things. As few people I can see they are new, and they want to learn this tool and molecular docking process with proper way. So you need to join earlier and be interactive. If you will not interact, you will not get anything from this workshop, I am telling you, because this is the new tool. You will not get this the videos of this tool anywhere. So better you will do practice hands on with me during this workshop. Okay, so thank you, student. I hope you people have enjoyed the today's session, and I hope you people have gained some knowledge.
Yeah, wait, let me get the second and third form. I just fill the first one. Let me just get the second and third form before you close, because if you close down, everything will disappear.
Yeah, I will send you in the official group also. The registered candidate
Okay,
will
okay.
get all the information. All the information you people will get in the official group. So don't worry. Registered candidate will be like premium candidate. So don't worry about
That's okay, thank you.
it. Okay.
That's okay.
This form is also for the student who have not registered. They have to fill and they have to submit, and the register and you have to do the registration. Okay. So thank you all for
Okay.
joining.
Thank you. Thank you so
If
much.
people have any feedback, you can give in this live session. It will be very beneficial.
So what time is tomorrow's class?
Tomorrow class will be around 6 p.m.
Okay, thank you.
Okay.
Thank you.
Please, student, be interactive. Please respond. That's the end of the class. If you have any doubt, kindly ask. Hello. Hello.
Thank you. I've been having network issues actually, and I was driving because I thought it was going to be evening today, but when I checked, it's actually afternoon in Nigeria.
Okay.
So I couldn't really follow up. Yes, I don't know if I can get a recorded version.
Okay. Okay. Uh, you will get the
Meanwhile,
recording of the day one, but for the
yes.
day two and other session, you need to register.
Okay. Meanwhile, I'm still finding it difficult to make payment. I was the one that was private shouting you about having issues with payments from Nigeria. Even
See,
my
Peter is
PayPal account, I was
from
trying to,
he's from Nigeria only. He have done the payment through PayPal.
huh?
Peter who have joined,
Yes.
he is from Nigeria.
I don't know why mine is giving me issues. I'll still try it this evening though.
You need to pay with your credit or debit card. Okay. After
Okay,
you
I will
just
try it
click on the
again.
link. Once the link will open, okay, then you have to select
Yeah.
pay with credit or debit card. When you click on the pay on debit card, you can just fill the details and you will be able to pay very easily.
Okay.
If
Okay.
you
I'll
cannot
try
able
it.
to pay, okay, if you cannot able to pay, if you have any issues in that, no problem, you just contact me again. We will see what we
Okay.
can do. Okay.
All right. All right.
Oh, thank
All
you.
right. Thank you very much. I hope to join tomorrow's session and get
Yeah, yeah,
everything.
sure.
Thank you.
Tomorrow will be around 6 p.m. Okay. So join early. Hello. Hello, Shruti or Shreya. Are you listening? Have you people listen this listen anything in this workshop? Can you confirm? Or you people have any doubt? Okay, you have any doubts, Shruti? Okay. So see you tomorrow. Join early and download all the files and keep in your working directory ready so we can proceed with the molecular docking process. Okay, and fill this form. Thank you, student. Thank you to thank you again for joining and listening this work session carefully. Thank you. See you tomorrow. Bye. You will get the recording by today.